Welcome to Tutorial Zone, where we simplify complex concepts. Before we dive deeper into this video, please make sure you subscribe, make sure you like, make sure you comment, make sure you share, and turn on your notification button so that every time I post a new video, you should be updated. This video is a continuation of the previous videos on the anatomy and physiology of the cardiovascular system, where we will specifically concentrate on platelets and hemostasis. I am Dr. Said Piri, so let us begin. At the end of this video or lecture, you should be able to do the following. You should be able to describe the structure, function, and origin of platelets. You should be able to define hemostasis and describe three mechanisms that contribute to hemostasis. These mechanisms include vascular spasm, platelet plug formation, and coagulation. In the previous videos, we looked at the anatomy and physiology of red blood cells as well as white blood cells. In this video, we're going to concentrate on platelets and hemostasis. The first thing that you need to understand about platelets is that during the process of hematopoiesis in the red bone marrow, these platelets developed from the myeloid stem cell. This myeloid stem cell differentiates into a megakaryoblast, and this megakaryoblast differentiates into a megakaryocyte. This megakaryocyte is a very large cell where about 2,000 to 3,000 pieces break off from this megakaryocyte, and these pieces become what are known as platelets. Talking about the structure of the platelets, these platelets do not have a nucleus, but they contain many vesicles. From the previous video under white blood cells, I mentioned that vesicles, you can think of them as small pockets or small plastic bags that contain materials in them. Some of the materials that can be present in these vesicles inside the platelet include ADP, they include thromboxane A2, serotonin, and other materials. These substances are very much important in the function of platelets, which is formation of a blood clot. When there is damage to the blood vessel to prevent bleeding, these platelets are going to help in closing off that damaged blood vessel so that the person should not bleed too much. In terms of their diameter, platelets are about 2 to 4 micrometers in diameter. When you talk about them in the total number of platelets are about 150,000 to 400,000 cells per microliter. If you compare them to red blood cells, you realize that red blood cells are more than platelets. In terms of their lifespan, platelets are about 5 to 9 days. If you compare them to red blood cells that are about 120 days, they live about 120 days in the circulation before they are broken down or before they are removed from the circulation. But platelets only live for about 5 to 9 days. This process of production of platelets, which is known as thrombopoiesis, is under the influence of the hormone, or is controlled by the hormone thrombopoietin. If you remember, the process of erythropoiesis was under the control of the hormone erythropoietin. So that is all about the structure of platelets. We can now turn our attention to hemostasis. First thing that you need to understand is that do not confuse this word hemostasis with homeostasis. Homeostasis is the maintenance of the relatively stable internal environment. There's a video on homeostasis on this YouTube channel. You can take a look at it. But our main focus is hemostasis, which means to stop bleeding or it is a sequence of responses that stop bleeding. So when there's a damage to the blood vessel, here is our blood vessel which has been damaged at this point here. Blood can start leaking out and this person can bleed to death. There could be loss of large volume of blood from the cardiovascular system, and this is known as hemorrhage. So the process by which we stop or we prevent hemorrhage, the process by which we stop bleeding, is known as hemostasis. And this process proceeds in 3 important steps or sequences. The first one is vascular spasm, followed by platelet plug formation, followed by blood clotting or coagulation. We can now look at vascular spasm, which is the immediate response that happens when there is a damage to the blood vessel. This vascular spasm is also known as vasoconstriction. It's also known as vasoconstriction. When a blood vessel has vasoconstricted, it means there is a decrease in the diameter of this blood vessel. What you have to understand before we talk about this vasospasm or vascular spasm is that a blood vessel has an epithelium on the inside, and this epithelium is known as an endothelium. It has endothelial cells. I'll just draw them on one side only, but there is an endothelium that is made up of endothelial cells on each side of an artery, arterial, or a blood vessel. So those are endothelial cells. These endothelial cells are sitting on a basement membrane, and this basement membrane is just on top of connective tissue that is made up of collagen fibers. So those are the collagen fibers separated from the endothelial cells by this basement membrane. Just underneath there, you also have smooth muscle cells. Those are our smooth muscle cells. And these smooth muscle cells are the ones that lead to vascular spasm. When these muscles contract, there is vasoconstriction that happens. So when there is a damage, you have a cut, for example, to this blood vessel, that damage and also release of some chemicals after damage, as well as a reflex in response to activation of pain receptors by that injury, is going to cause contraction of these smooth muscles. And when these smooth muscles contract, a blood vessel that was big like this in diameter becomes smaller in diameter. That is vasoconstriction or vascular spasm. This is important in that it reduces the diameter of that blood vessel and therefore reduces the flow of blood to that injured area, so that now we should have less blood going to that injured area, preventing loss of a lot of blood to be lost. So this reduces the blood loss, and this mechanism happens within several minutes to several hours, so that there is time for other hemostatic mechanism to go into operation. The next mechanism after vascular spasm is platelet plug formation. This process or mechanism happens in 3 steps or stages: platelet adhesion, platelet release reaction, and platelet aggregation. So when the blood vessel is not damaged, blood is flowing freely and platelets are also flowing in this blood vessel without sticking to the endothelium. However, damage of the endothelial lining or the endothelium of the blood vessel leads to exposure of the collagen. So remember, we have these endothelial cells here, like that, and these endothelial cells are sitting on a basement membrane, like that, and then underneath there we have the collagen. We have the collagen fibers. If you look at this diagram here, you can see these collagen fibers just underneath these endothelial cells, that collagen there underneath the endothelial cells. So the damage to a blood vessel exposes these collagen fibers under the endothelium. So once these collagen fibers have been exposed, the platelets now that are flowing in this blood vessel come and stick, or they come in contact and stick to these collagen fibers that are underneath the endothelial cells. So the process by which platelets stick to the damaged endothelial cells or to the damaged endothelium, they stick to the collagen fibers and damaged endothelium, is known as platelet adhesion. After the platelets have adhered, what happens next is platelet release reaction. So when you look at the structure of a platelet, it is kind of irregular and disc shaped. But when these platelets attach to the collagen and damaged endothelium, they change shape in that they become activated and change shape, and they look spiky like that. They have spikes on their surface like that. So if you look at these platelets, they are normal in terms of shape, but once they have attached to the damaged endothelium and the collagen, they become spiky like that. So they become activated and they release chemicals that are present in the vesicles, and some of these chemicals are ADP, thromboxane A2, as well as serotonin. ADP contributes to activation of more platelets. We're going to talk about later on. Thromboxane and serotonin also, they are vasoconstrictors. And these vasoconstrictors, they contribute to vascular spasm. So that now the blood vessel should decrease more in diameter. There should be more blood, there should be less blood now going to that damaged area to prevent blood loss. The ADP that has been released, the thromboxane that has been released, also contribute to activation of nearby platelets. So they go to nearby platelets. So this process by which these platelets get activated and release these chemical substances present in the vesicles is known as platelet release reaction. Then the next thing is platelet aggregation. The ADP, the thromboxane, they activate nearby platelets. So if these are the nearby platelets that are not activated, it activates them and make them sticky, so that now they can come and attach to these platelets that were already present at that damaged site. And these platelets that are newly activated, they come and attach further and further, just like that. More platelets are coming in, attaching to the old ones that were already there. So this process by which newly activated platelets attach to old platelets that were present at the damaged site is known as platelet aggregation. And once that has happened, this mass or this group of platelets that is present at the damaged area of a blood vessel is known as a platelet plug, or a primary platelet plug, or hemostatic plug. In some cases where the damage is too small to a blood vessel, this platelet plug is enough to cause stoppage of bleeding. However, in larger vessels, this plug is too loose. In larger damages, where the damage is very big, this plug is too loose to cause a stoppage in the loss of blood. So what we need to do is we have to come and add something to strengthen it, and that is going to be the fibrin threads, and we're going to look at it under the coagulation cascade. So when you have a normal blood vessel, blood in this normal blood vessel tends to be maintained in a liquid state. However, when there's a damage to the blood vessel and this blood is exposed to the outside of the body, there is formation of what is known as a gel or a blood clot. So this blood clot is simply just the formed elements, which are the RBCs, the platelets, and the WBCs that are trapped within threads, the fibrin threads. They are trapped within the fibrin threads. So this blood clot separates from the liquid component of the blood. So that liquid component of the blood that comes out of a blood clot does not have the clotting proteins. It doesn't have fibrinogen in it. Remember, fibrinogen is very important for clotting. So it doesn't have fibrinogen in it, and this fluid component of blood that does not have clotting proteins in it, that does not have fibrinogen in it, is known as serum. So that's the difference between serum and blood plasma. Blood plasma has all the three proteins: albumin, fibrinogen, and globulins. But serum does not have this fibrinogen. The process by which this blood clot or this gel is formed is known as clotting or coagulation. And we are going to talk about coagulation in a while. It leads to formation of what are known as fibrin threads. This whole process of coagulation depends on several factors which are known as clotting factors or coagulation factors. We have 12 coagulation factors in total, from 1 all the way to 13. What you need to understand is that there is no factor 6. These coagulation factors are not arranged in a sequence by which they're not arranged in these numbers based on how they act in the coagulation cascade or during coagulation. They're just arranged like this based on which one was discovered first. Most of these factors are produced by the liver. So there are 3 sources of these coagulation factors. Majority come from the liver cells, which are the hepatocytes, and then some of them come from the platelets. Some of them come from the damaged tissues. When the blood vessel is damaged, even the tissues surrounding those blood vessels are also going to be damaged. So those damaged tissues can also release some coagulation factors, but majority come from the liver cells. The other important point to understand is that there are 4 coagulation factors that depend on vitamin K availability for their synthesis or production. That is factor 2, factor 7, factor 9, and factor 10. So what this means is that if an individual has a deficiency in vitamin K, they'll have a deficiency in these coagulation factors, and therefore they're going to have bleeding tendencies. They'll have challenges forming a blood clot or forming fibrin threads that are going to stabilize their clot. So they are most likely at an increased chance of bleeding a lot. The process of coagulation or blood clotting happens in 3 stages. We have the extrinsic pathway, the intrinsic pathway, and the common pathway. The extrinsic pathway happens or is initiated because of materials, let's say clotting factors, leaking into the blood vessel. So if this is our blood vessel that has been damaged here, remember you can also have damage to the tissue. So the tissues on the outside of the blood vessel have been damaged. So materials that initiate this extrinsic pathway leak from the damaged tissues into the blood vessel. But for the intrinsic pathway, the materials that initiate, or the clotting factors that initiate the events in the intrinsic pathway, are present within the blood vessel itself. They are present within the blood vessel itself. So once these materials or clotting factors come in contact with the damaged endothelium or the collagen fibers, the intrinsic pathway, or the sequence in the intrinsic pathway, the events in the intrinsic pathway will be initiated. The common pathway, on the other hand, is the convergence point. It's a convergence point for both the intrinsic and the intrinsic pathway. So we have extrinsic pathway this side, intrinsic pathway this side. All these pathways will lead to production of what is known as prothrombinase, an enzyme known as prothrombinase. Once this enzyme has been produced, it is going to go into the common pathway. The goal in the common pathway is to produce insoluble fibrin threads, and these fibrin threads are very much important in stabilizing the platelet plug. Remember, we produced a platelet plug in the previous explanations. That platelet plug wasn't stable, but now we need to put threads on top of the platelets and other form them. So if these are the platelets, we want to put the fibrin threads here on these platelets so that we can stabilize this clot. The main goal of the common pathway is to produce these fibrin threads. So this is what I was talking about, the extrinsic and the intrinsic pathway. Their common goal is to produce an enzyme known as prothrombinase. And this prothrombinase is going to be used in the common pathway, which is the convergence point, the point where both the intrinsic and the intrinsic pathway meet. And what do we want to do in the common pathway? We want to produce an enzyme thrombin. So it is the prothrombinase that is going to be used to produce an enzyme thrombin. This thrombin, the enzyme thrombin, is going to be used to form the insoluble fibrin. Once that insoluble fibrin threads have been formed, they're going to be used to stabilize the blood clot. I have mentioned in the previous slides that the extrinsic pathway is named it is named as the extrinsic pathway because the material or the clotting factor that initiates this pathway, which is known as the tissue factor or thromboplastin, leaks into the blood vessel from the tissues that are present outside the blood vessel. It comes from the outside, so it's extrinsic to the blood vessel. So we call this the extrinsic pathway. So there are three things that happen in the extrinsic pathway. The first thing is leakage of the tissue factor into the blood vessel. The material is coming out of the blood vessel. The clotting factor, which is thromboplastin or tissue factor, abbreviated as TF as we move ahead, comes from the cells that are present outside the blood vessel. Once those cells or tissues have been damaged, they release this tissue factor into the bloodstream. Once that tissue factor is in the bloodstream, the second thing that should happen is activation of clotting factor X or factor 10. Once this factor 10 has been activated, there will be a sequence or a series of events that will lead to the formation of the enzyme prothrombinase. What you need to understand is that majority of these clotting factors or these enzymes that are present in this coagulation cascade, they are located, or they are present in an inactive form. They are inactive. But this series of events, or this sequence of events that happen, is one clotting factor activating the next until we form insoluble fibrin threads. In the extrinsic pathway, three things happen: initiation, which is because of the leaking of the tissue factor into the blood vessel; activation of factor 10; formation of the enzyme prothrombinase. In general, that's what happens in the extrinsic pathway. Now, how are these events happening? The first thing is, when there's damage to a blood vessel, there's also tissue trauma. There's also damage to the surrounding tissue. And those cells in the tissue that have been damaged release tissue factor, or thromboplastin. This thromboplastin enters the bloodstream. And in the bloodstream, this tissue factor, in the presence of calcium, activate factor 10. So factor 10 was inactive, and now it has been activated. This activated factor 10 combines with factor 5 in the presence of calcium to form the enzyme prothrombinase. So the events that happen in the extrinsic pathway in comparison to the intrinsic pathway are kind of short. But what I want you to understand or to remember is that the common goal between intrinsic and extrinsic pathway is formation of the enzyme prothrombinase. So you can see that once tissue factors leaked into the bloodstream, the events in the extrinsic pathway were initiated, where the tissue factor in presence of calcium activated factor 10, and then activated factor 10 combined with factor 5 in the presence of calcium to form the enzyme prothrombinase. So this is how the extrinsic pathway happens. We can now talk about the intrinsic pathway. From the previous slides, I've mentioned that the intrinsic pathway is named so because the clotting factors that initiate this pathway are present within the blood, or they are contained within the circulation. So they are intrinsic to the blood vessel, they are within the blood vessel. So what they require for them to initiate this pathway is an endothelium that has been roughened or that has been damaged. So there's exposure of collagen fibers present under the endothelial cells. Even the endothelial cells themselves have been damaged. Once that has happened, there should be activation of clotting factor 12. In this case, it's factor 12, not factor 10. It is factor 12 now that leads to a series or a sequence of events or reactions where factor 10 is activated. So from factor 12, we're going to activate factor 10, and this activated factor 10 now should combine with factor 5 in the presence of calcium to produce the enzyme prothrombinase. If you remember, I mentioned that both the extrinsic and the intrinsic, their main goal is to produce the enzyme prothrombinase. So even here, the events might look different with the extrinsic pathway, but their main goal is to produce the enzyme prothrombinase. Here are the details of how the events happen in intrinsic pathway. So the first thing that we need to do is, since these clotting factors are present within the blood vessel, we need damage to the endothelial cells and exposure of the collagen fibers, as well as the damage to the platelets. So once that has happened, this damage to endothelial cells, exposure of collagen factors, the platelets have been activated and some have been damaged, they're going to lead to the activation of factor 12. Activated factor 12 in the presence of calcium, as well as some of the platelets phospholipids. So these activated platelets also release some phospholipids. So together, activated factor 12 and calcium and these platelets that come from the phospholipids are going to activate factor 10. Once they activate factor 10, they're going to lead to the activation of, activated factor 10 is going to combine with factor 5 in the presence of calcium to form prothrombinase. So there are going to be series of events, but all these series of events or this sequence of events lead to the production of activated factor 10. Once factor 10 has been activated, it gets to combine with factor 5 in the presence of calcium to form prothrombinase. So now we have seen how the intrinsic pathway happens. The common pathway was the convergence point. It's where the extrinsic and the intrinsic pathway meet after producing the enzyme prothrombinase. So prothrombinase, or the formation of prothrombinase, marks the beginning of the common pathway. What happens now is we have an inactive fibrinogen molecule. So fibrinogen here, you can see he's very sad, he's inactive, right? We are going to use this prothrombinase to activate this fibrinogen into loose fibrin threads. So now the fibrin has been activated, smiling there like that, and these fibrin threads now can go under, they are laid on top of the platelets that we have in the damaged endothelium, but they are very loose in that they cannot hold the platelet plug. What we need now is to stabilize these fibrin threads by activating factor 13, which is fibrin stabilizing factor. This fibrin stabilizing factor is released by platelets. It's present within the platelets. So it's the platelets that releases them. So that's what the common pathway is all about: production of fibrin threads and activation of factor 13 and so on. So this diagram is showing you what happens in the common pathway. Remember, we have produced prothrombinase. What does this enzyme do in the presence of calcium? This prothrombinase converts prothrombin to thrombin. This thrombin now, in the presence of calcium, acts on the inactive fibrinogen and converts it to loose fibrin threads. You can see these fibrin threads, they are not so strong, they cannot hold the clot for too long. What should happen now is we need to strengthen them. We need to stabilize these fibrin threads. What do we do is, there's going to also, this thrombin that we have produced is also going to lead to activation of factor 13, which is a fibrin stabilizing factor, and once it has been activated, it is going to act on the loose fibrin threads, converting them to strengthened fibrin threads that can withstand or that can hold this clot for longer. So you can see from this diagram what happens to the fibrin. You can see here they are looking so small and loose. There they are looking more stronger now in the intermediate stage, but after they have been stabilized, you can see they have trapped those red blood cells, they have trapped the platelets under them. The formed elements are trapped within these fibrin threads. So this is what happens in the common pathway. We want to produce these strong fibrin threads that can hold the clot, they make it sturdy, they make it strong. So once this clot has been formed, these fibrin threads are now laid on the platelets as well as other formed elements. They are trapping these platelets and the formed elements. What happens is that these platelets will start pulling these fibrin threads, and as these fibrin threads are being pulled, what happens is that the clot starts to contract or to tighten as it pulls the ends of the damaged blood vessel. So this is what clot retraction or consolidation or tightening means. It's just that the threads are tightening and there's pull of these damaged ends, or there's pull of these two ends of the damaged blood vessel, which is going to stabilize the clot even further. As these threads are pulling, the fluid starts escaping this clot, and this fluid that escapes this clot is known as serum. You have to understand that these formed elements, the RBCs, platelets that have been damaged, that have been trapped here, are not going to escape. The other thing that you have to understand is fibrinolysis. So once the clot has been formed, it has a tendency of increasing in size, and this can be dangerous in that it can end up blocking the circulation. It can end up closing the lumen of the blood vessel. So as a way of preventing this from happening, there's a protein plasmin or fibrinolysin that tends to break down these unwanted small clots to prevent them from becoming larger and larger, so that they shouldn't block the circulation. So that's what fibrinolysis is all about. We have now come to the end of this lecture. In this video, we have discussed the structure and function of platelets. We have also discussed hemostasis and the mechanisms of hemostasis. We have looked at vascular spasm. We have looked at platelet plug formation. We have also looked at the coagulation, extrinsic, intrinsic, and common pathways. Please make sure that you watch part 1, which is Introduction to Blood and Red Blood Cells. Make sure you watch part 2, which is a focus on white blood cells, before you watch this part 3, a focus on platelets and hemostasis. Please make sure you subscribe, make sure you like, make sure you share, and turn on your notification button so that every time I post a new video, you should be updated. I am Dr. Said Piri. Till next time.